Biology of bitterness. To many people, grapefruit is palatable only when doused in sugar. Bitter blockers like adenosine monophosphate could change that. There is a reason why grapefruit juice is served in little glasses. Most people don't want to drink more than a few ounces at a time. Naringan, a natural chemical compound found in grapefruit, tastes bitter. Some people like that bitterness in small doses and believe it enhances the general flavor, but others would rather avoid it altogether. So juice packages often select grapefruit with low naringan, though the compound has antioxidant properties that some nutritionists contend may help prevent cancer and arteriosclerosis. It is possible, however, to get the goodness of grapefruit juice without the bitter taste. I found that out by participating in a test conducted at the Linguagen Corporation, a biotechnology company in Cranbury, New Jersey. Sets of 2 miniature white paper cups, labeled 304 and 305, were placed before 5 people seated around a conference table. Each of us drank from one cup and then the other, cleansing our palates between tastes with water and a soda cracker. Even the smallest sip of 304 had grapefruit's unmistakable bitter bite. But 305 was smoother. There was the sour taste of citrus but none of the bitterness of naringan. This juice had been treated with adenosine monophosphate, or AMP, a compound that blocks the bitterness in foods without making them less nutritious. Taste research is a booming business these days, with scientists delving into all 5 basics: sweet, bitter, sour, salty, and umami, the savory taste of protein. Bitterness is of special interest to industry because of its untapped potential in food. There are thousands of bitter-tasting compounds in nature. They defend plants by warning animals away and protect animals by letting them know when a plant may be poisonous. But the system isn't foolproof. Grapefruit and cruciferous vegetable like Brussels sprouts and kale are nutritious despite and sometimes because of the bitter-tasting components. Over time, many people have learned to love them, at least small doses. Humans are the only species that enjoys bitter taste, says Charles Zucker, a neuroscientist at the University of California School of Medicine at San Diego. Every other species is averse to bitter because it means bad news. But we have learned to enjoy it. We drink coffee, which is bitter, and quinine in tonic water too. We enjoy having that spice in our lives. Because bitterness can be pleasing in small quantities but repellent when intense, bitter blockers like AMP could make a whole range of foods, drinks, and medicines more palatable and therefore more profitable. People have varying capacities for tasting bitterness, and the differences appear to be genetic. About 75% of people are sensitive to the taste of the bitter compounds phenylthiocarbamide and 6N-propylthiouracil, and 25% are insensitive. Those who are sensitive to phenylthiocarbamide seem to be less likely than others to eat cruciferous vegetables, according to Stephen Wooding, a geneticist at the University of Utah. Some people, known as supertasters, are especially sensitive to 6N-propylthiouracil because they have an unusually high number of taste buds. Supertasters tend to shun all kinds of bitter-tasting things, including vegetable, coffee, and dark chocolate. Perhaps as a result, they tend to be thin. They're also less fond of alcoholic drinks, which are often slightly bitter. Dewar's Scotch, for instance, tastes somewhat sweet to most people. But a supertaster tastes no sweetness at all, only bitterness, says Valerie Duffy, an associate professor of dietetics at the University of Connecticut at Storrs. In one recent study, Duffy found that supertasters consume alcoholic beverages, on average, only 2 to 3 times a week, compared with 5 or 6 times for the average non-tasters. Each taste bud, which looks like an onion, consists of 50 to 100 elongated cells running from the top of the bus to the bottom. At the top is a little clump of receptors that capture the taste molecules, known as tastants, in food and drink. The receptors function much like those for sight and smell. Once a bitter signal has been received, it is relayed via proteins known as G proteins. The G protein involved in the perception of bitterness, sweetness, and umami was identified in the early 1990s by Linguagen's founder, Robert Margolsky, at Mount Sinai School of Medicine in New York City. Known as gustjuicin, the protein triggers a cascade of chemical reactions that led to changes in ion concentrations within the cell. Ultimately, this delivers a signal to the brain that registers as bitter. The signaling system is like a bucket brigade, Margolsky says. It goes from the G protein to other proteins. In 2000, Zucker and others found some 30 different kinds of genes that code for bitter taste receptors. We knew the number would have to be large because there is such a large universe of bitter tastants, Zucker says. Yet no matter which tastant enters the mouth or which receptor attaches to, bitter always tastes the same to us. The only variation derives from its intensity and the ways in which it can be flavored by the sense of smell. Taste cells are like a light switch, Zucker says. They are either on or off. Once they figured out the taste mechanism, scientists began to think of ways to interfere with it. They tried AMP, an organic compound found in breast milk and other substances, which is created as cells break down food. AMP has no bitterness of its own, but when put it in foods, Margolsky and his colleagues discovered, it attaches to bitter taste receptors. As effective as it is, AMP may not be able to dampen every type PF bitter taste, because it probably doesn't attach to all 30 bitter taste receptors. So Linguagen has scaled up the hunt for other bitter blockers with a technology called high-throughput screening. Researchers start by coaxing cells in culture to activate bitter taste receptors. Then candidate substances, culled from chemical compound libraries, are dropped onto the receptors, and scientists look for evidence of a reaction. In time, some taste researchers believe compounds like AMP will help make processed foods less unhealthy. Consider, for example, that a single cup of Campbell's chicken noodle soup contains 850 milligrams of sodium chloride, or table salt more than a third of the recommended daily allowance. The salt masks the bitterness created by the high temperatures used in the canning process, which cause sugars and amino acids to react. Part of the salt could be replaced by another salt, potassium chloride, which tends to be scarce in some people's diets. Potassium chloride has a bitter aftertaste, but that could be eliminated with a dose of AMP. Bitter blockers could also be used in place of cherry or grape flavoring to take the harshness out of children's cough syrup, and they could dampen the bitterness of antihistamines, antibiotics, certain HIV drugs, and other medications. A number of foodmakers have already begun to experiment with AMP in their products, and other bitter blockers are being developed by rival firms such as Cenomix in La Jolla, California. In a few years, perhaps, after food companies have taken the bitterness from canned soup and TV dinners, they can set their sights on something more useful, a bitter blocker in a bottle that any of us can sprinkle on our Brussels sprouts or stir into our grapefruit juice.